So I think we're going to resume for the last talk of the day, not the least one. We are very pleased to have Eric Wieschaus. I mean, I don't think I have to introduce Eric. I mean, everybody knows what he has done and what he has received in 1995. 1995, was it? Yeah. With Ed Lewis and Nuslan Vollard. I think that what he's done since is in the continuation, more or less, of this initial work on the development of the fly, I mean, the fly embryo, with a lot of things also on cellularization and what happens after cellularization, during the gastrulation, and all these movements and how cells take shape and, well, you know, life. Well, so Eric, I'm very pleased that you accepted to come, and we are all ears.
I think we could have the lights down a little bit, but not much more than that. That's fine. So what I'd like to do is talk this afternoon about maternal gradients and transcriptional responses in the Drosophila embryo. And this is, in the broader sense, the idea of maternal gradients or gradients in general and how cells respond to those gradients to make developmental choices. Of course, a general problem in all of developmental biology. It's, as was pointed out this morning, I think it's useful to think about the process in terms of decision-making, this decision-making event essentially involving some input information, in this case, maternal morphogen gradients, that through some process that we don't fully understand results in an output, which is precise transcriptional activation that determines cell fates. The important idea here that I'd like to get across in this talk is the concept that associated with each of these two terms is a input and output is an information content. Information is supplied by the maternal gradients, and in some way that information is used to generate a new form of that information, which is the output that controls cell fates. Now, the particular system that I have worked on since I was as young as many of the people in this room is the early Drosophila embryo. And over the past 30 years, we've learned quite a bit about this. This is one aspect of the maternal patterning is established by the localization of a maternal RNA called bicoid, the anterior end of the egg. And that RNA, which is deposited during oogenesis and inactive until the egg is fertilized, is then translated into a protein which diffuses from this source to establish a concentration gradient of the protein. Bicoid is a homeodomain protein. As far as we know, it functions as a transcriptional activator. And downstream of this maternal input are a set of zygotically active genes that define different regions of the embryo. They can be arranged both in terms of their pattern, in large domains, increasingly smaller domains, the gap genes, parallel genes, polarity genes, but also in terms of genetic experiments into a hierarchy of functions that ultimately in some way connect the information of this bicoid gradient into refined patterns of gene expression that are actually distinct in single rows of cells. Now, I thought it would be useful just to review a little bit of our transformation of thinking about this process over the couple years. Although when Christian and Nuslan and Vohard described this system, we focus pretty much on the concept of genetic hierarchies. Very quickly, those genetic hierarchies became temporal stages and this is a quote from Hans Meinhardt, the formation of segments begins with a simple pattern, the gradient, and reaches the complex pattern by passing through a hierarchical series of intermediate patterns. So there's a temporal sequence here. Very quickly, that hierarchical pattern, the interactions were between the genes that resulted, presumably, in this final pattern, were associated with a systems biological approach with robustness. This is a quote from a famous textbook in the United States by Bruce Alberts. At each stage in the sequence, new signals come into play, providing substantially localized differences of concentration to define new details in the pattern. Sequential induction is thus a robust process, and the idea is to couple this sequence of interactions to a set of processes which is therefore inherently more robust against noise. The system works reliably to produce fly embryos that have all have the same pattern, despite the essential imprecision of biological control systems. This arguing that there is some essential aspect of biology that is imprecise. And then in another further development of this idea is that the patterning here being an emergent system is differential bindings from a paper from Steve Small's lab. Differential binding sensitivity of bicoid is not the primary design principle that positions bicoid-dependent expression targets. The bicoid gradient can establish only a rough framework that is elaborated by interaction with zygotic segmentation genes. I think this view is very close to what we could think of as a kind of a standard textbook, not only for this process, but for much of how we think about developmental biology, that processes, there are initial cues or signals that set off interactive systems that eventually, through their interactions with each other, generate complex patterns. Characteristics of this kind of view is that the maternal gradients would provide only general cues to a system that's inherently self-organizing. I mean, down here, the interactions will self-organize, and what you're doing, you're providing a cue or a bias to orient the organization process. The input information is low, and it varies from one embryo to the next. So the expectation is that the actual gradient is sufficiently variable that really all that it can provide is general cues. Information complexity then increases at each step, and consequently, you expect very little correlation between the initial input, whatever it is in a given circumstance, and the final detailed response of the cells. And this is a view, I think that's shared by most developmental biologists. I have to say it was also my view in thinking about the process initially over the years. Actually, it relates back, and I deleted this slide, but it relates back to some famous quotes from Theodore Boveri at the beginning of the previous century in the 1902 paper, where he argues that a paper that's directed at the role of chromosomal activities in early embryonic development, where he argues that maternal information in the cytoplasm must be very minimal and very simple, and all of that information is then elaborated on by the genetic activities of the individual chromosomes. So my own sense of this process began to be altered by work that Thomas Greger did as a graduate student at the time, where he labeled and presented careful measurements of the bicoid gradient in living embryos, taking care with orientation, with staging. And what he showed, and this was quite remarkable at the time, was that unlike most of us had expected, the bicoid gradient was in fact extremely reproducible from one embryo to the next. That the concentrations at a given position along the anterior-posterior axis varied by less than 10%, and that the concentrations on average vary by less than 10%, and the differences between neighboring, the variability was also in the range of 10%, which means that effectively cells could identify their position and distinguish their position reliably from their nearest neighbor, or that would be a 1% accuracy along the length of the embryo. This is remarkable precision for a gradient. It may just simply reflect the facts of biophysics, that if you put a certain amount of RNA into the egg, translate it at a certain amount, and allow it to diffuse at a certain rate and degrade at a certain rate, it will produce a reproducible gradient. The more complicated question, though, is really the Bicoid concentration, therefore, is actually sufficient to establish distinct reproducible cell fates in neighboring cells. But that would be true only if cells are able to use or access that information. So that's really going to be the theme of my talk today, whether and how it is, whether cells can access the information inherent in the Bicoid gradient, and if so, what are the processes involved that allow the cells to do that? Now, the big problem with most of these studies, and I think this is a problem in general in biology, certainly in developmental biology, is measurement and establishing strategies that allow us to distinguish, and when we're looking at, since information depends on reproducibility of molecular distributions, and therefore, what one has to be able to do is to establish measurement protocols that allow you to distinguish between those aspects of variability which are inherent to the biological system and therefore inherently decrease information content from those which are imposed by the experimenter, either by the variable stages that one's collecting the data from, variable orientations of embryos, variable microscope setups. And I think that one of the critical feature in my own lab and working procedures has been becoming increasingly conscious of the need for all of us to deal with those sources, deal with the sources of variability in establishing measurements. So the question that we'd like to address is how accurately can cells read gradients? And is positional information that we've argued that is present in the initial maternal gradient, is it lost in the transition, say, between maternal gradients and the first gap gene expression responses? And what I'd like to present for you first in this talk is a system which was we developed at Princeton, essentially a Bayesian system for establishing lookup tables for embryonic patterning. And I'll explain what that means. This is the work of Julian Dubuis, Mariela Petkova, Gaspar Tacek, Thomas Greger, me, and Bill Bialek. And essentially, the first step in this procedure was to develop a set of antibodies to all 4 gap genes in different animals such that we could individually measure the gap gene distributions in single embryos for all of these, for all 4 genes in single embryos, quantify them with the care to measurements at given stages during development. And then what this data gives us is at a given position along the anterior-posterior axis of the embryo, we have a certain probability that a given one of these genes will be expressed, either at very low probability or at very high probability. That's the probability. What we would like to know, and what the embryo would like to know is, from an information standpoint, is what is the probability that if this information, this gap gene expression is going to give you information about position, what the embryo has to be able to do is to convert this information into the probability that I'm at position X, given that I have a set of gap gene, given that I have these concentrations of each of these 4 gap genes. And using these formulas and joint probability distributions on literally hundreds or thousands of embryos carefully staged during cycle 14. What Mariela Petkova has done, I'm just summarizing this figure here, is something like a lookup table which summarizes the quantitative distributions of individual gap genes at given points along the anterior position, and the computer's estimate of where that measurement was taken from in the embryo. The width of this line, the fuzziness of the line, indicates the variability and inaccuracy of the estimates here. One thing you can see is that the information is basically enough to define all positions along the anterior posterior axis, along this diagonal, and with a certain variability or fuzziness. We can then ask if individual positions here, we can show that an individual set of gap genes association correlates with and allows us to predict, correlates with a defined position, the certain variability, and allows us to predict the expression of parallel genes 8 to 10 minutes after that particular measurement was made. So we know that this correlation tells us that this information in gap genes is in some way associated with parallel genes, with parallel gene expression. Now, once we've established this technique, which this particular image is from embryos collected at 43 minutes, plus minus 2 or 3 minutes in cycle 14, we can go back and say, follow the same protocols on embryos throughout all of cycle 14. Prior to cycle 14, these gap genes are not expressed as proteins, and we believe they have very little function. The next slide shows you that data, and one thing that's striking when you look at embryos is that over the period of time from 10 to 15, 20 minutes, 45 minutes, the gap gene expression patterns change substantially, as you can see here in these figures. But what's interesting is if you carefully define the stages of your embryos, it's possible even from the earliest stages to build tables that substantially, although not entirely, define position. So here's an area in the embryo where the position is still somewhat ambiguous at this hours, but you can see over time is corrected. In the region of the embryo, though, where parallel genes are expressed, what's striking to me is that even at the earliest stages, at 10 minutes into cycle 14, the positional information is very precise and sufficiently precise to define or predict parallel gene expression. We can make these predictions more quantitative by defining a property we call positional error, which is essentially the fuzziness or the width of the standard deviation divided by the length. And what we can see is that even throughout all of these stages, the errors are somewhere between 1.5% egg length down to 1% egg length. So there's very little change, very little increase in information content in embryos during the progression through cycle 14. This, in my mind, argues that the system is not loaded with a lot of noise early that has to be corrected by interactions between different gap genes, but that somehow the genome is able to respond to these concentration. The concentration differences are the input information is constant, or not constant, but information content is constant, and the cell's ability to respond to that information is constant. This suggests that cells are able to read information down to establish precise positions with about 1% accuracy from the earliest stages, and probably, in my mind, with very little evidence for interactions between these genes to produce pattern. Now, once we've established the system, we can begin to use it, and we have begun to use it, to examine other aspects of the information transfer. Many of you know that the maternal information in Drosophila, it involves 3 different patterning systems: the bicoid system that I talked about and that we've already talked about this morning, and 2 other systems, the receptor tyrosine kinase torso signaling system with the associated transcription factor Capicua, and the nanos localized RNA translational control system with its associated transcription factor maternal hunchback protein. And all 3 of these systems are known to be contribute to patterning in the embryo. What we can do now, though, is ask if one changes the information content by removing either the torso system, the nano system through moving out, or moving bicoid, what is the consequence on cell decisions? And what you can see here for the typical case for torso is that the signaling, the gap gene expression patterns that are generated in the embryo deviate from the standard diagonal at the anterior and posterior ends of the embryo, but along the middle of the embryo, the patterning tracks very well. Similarly, with Oscar, the patterning tracks well. The anterior end, and there's a region where there's no tracking. What's interesting and striking for us when you remove bicoid, and this is different from many of the cases, is that at all points along the anterior-posterior axis, the patterning and decisions, the positional evaluations are altered when you alter bicoid. And what this suggests is that all anterior-posterior position along the all positions along the anterior-posterior axis use bicoid to establish its position. We can do a variant of this experiment, and instead of removing single components, we can reduce the embryo down to having only a single maternal component and ask how well does the embryo pattern in the absence with using any one of these systems. And what you can see with no torso patterning and no Oscar patterning, we get difficult to interpret signals that deviate over their entire length from the expected diagonal. And yet embryos that have only bicoid, that is hunchback nanos torso-like germline clones, have patterns that essentially track the basic patterning in the embryo through along most of its length, although there's still deviations at the anterior and posterior pole. And so information from bicoid alone is sufficient to track much of the wild-type pattern. Now, I'd like to just reiterate by presenting the same data in a slightly more visual fashion, that how much can bicoid do on its own? And again, the strategies that we have to use here is to not only remove torso-like and nano signaling, but to remove hunchback, which is an inhibitor of abdominal patterning formation in germline clones, in torso-like females. This is my major accomplishment of the past 3 years in the lab with my own dirty little hands. And so that's why I'm spending a lot of time on it in this talk. You have to realize that's the way things are. This is wild type. This is the embryos that have only bicoid. They have head structures. They're missing certain terminal head structures. They're missing the most terminal structures here, but they pattern all the way out to the 6th abdominal segment. And if we know that this is due to bicoid, because if we generate embryos that are all the same genotype of embryos that are also missing bicoid, all patterning goes away. So this is what bicoid can do on its own. And that's interesting because if we then extrapolate back to the shape of the gradient, what this is or look at the embryos where we see knirps and giant expression in these embryos, it means that these patterns are being established out in regions of the embryo where the changes in bicoid are really very small, and suggests that the embryo is probably, or nuclei are able to read bicoid concentrations at least as well as we are the microscope. Now, one possibility, though, this tells us that bicoid is directly or indirectly responsible for these patterns. One possibility is that these patterns are dependent, that they are in fact dependent on gap gene interactions. One simple model would be that bicoid, which was known to activate hunchback, might activate hunchback, hunchback might activate cripple, cripple might activate knips, knips might activate giant. So you'd have a relay system where the pattern would really only reflect bicoid's activity at the anterior end. One of the ways that we're beginning to test that is to genetically manipulate the zygotic content of the embryos, and only the embryos. I'll present one result for you, which is when one eliminates the hunchback gene, which would be the major player here. And what you can see is that in the absence of maternal and zygotic hunchback, and nanos, and torsal embryo patterns, and the abdominal patterns are cripple, cnips, and giant are still established in the regular order. So all of this argues, in my mind, that bicoid, as a single transcription factor, may directly activate with great precisional accuracy the boundaries of all of these gap genes. That raises the next question. How does a single maternal transcription factor govern multiple cell fates? As I mentioned, bicoid is graded along the anterior-posterior axis of the embryo. One simple model, and it has been the attractive model over the past years, has been that you have a gradient of a transcription factor which activates transcription. You can imagine that individual genes that respond to that system have enhancers. The anterior targets would have low affinity such that bicoid protein only at very high concentrations would be able to bind to and activate those genes. Similarly, posterior targets would have very high affinity. And can this be tested? Many laboratories have set out to try to test this over the past years. It's been very difficult, I have to say, to demonstrate using the in vitro techniques for affinity or binding of analyses of enhancers to be able to test this possibility. And I think that to do that, what we need to do, basically need to do two things. We can't just focus on gap genes. We need to have measure direct interactions between bicoid and as large a number of target enhancers as possible. And we need to do those measurements in vivo, in embryos, rather than in vitro. And these are the experiments that Colleen Hannon and Shelby Blythe carried out in the lab and have just been published in eLife, essentially analyzing the nature of bicoid transcriptional responses. Right. So basically, the strategy that they used was a kind of enhanced bicoid chip experiment that involved visually hand staging embryos 10 to 15 minutes into cycle 14. And what is interesting when you do this is that you identified, and they identified 1,027 sites in the genome that reproducibly under all conditions bind bicoid. And this bicoid binding is, interestingly, for these 1,027 targets that were established simply because of the magnitude of the bicoid binding effect, bicoid binding is independent of the presence or absence of any of the other maternal systems or the transcription factors that they govern. So we think what we're looking at here is just direct bicoid interactions with enhancers, and the role of these other systems must in some way operate downstream of physical binding. Now, these are binding sites. They aren't necessarily enhancers. To address that issue, what we did was take advantage of a collection of reporter constructs that Alex Stark has generated, where he's basically taken random genomic fragments of about 2 kb, fused them to reporters, and then ask whether these reporters, this is a database that's available in Furfly, whether these reporters drive, whether these fragments of the genome will drive expression, transcription, and then when and with what pattern. And if you take random reporters from this collection, about 7% of them drive expression at the blastoderm stage. If we look for that fraction of this reporter collection that contain binding sites that we measure for bicoid, somewhere between 50 and 60% of these fragments will drive expression. And interestingly, they drive expression in complicated anterior patterns that are all different from each other. What this suggests is that the collection of enhancers, that these DNA binding sites, largely may function as enhancers to drive expression of adjacent genes. Now, the next step is you have a set of regions which bind bicoid protein. What you would like to know to test concentration dependence is, do they all bind with the same affinity? This is now getting to the idea of developing an in vivo affinity assay for binding. And the strategy that Colleen developed was to generate transgenic lines of bicoid that remove the 3' UTR and produce a bicoid RNA, which is now uniformly localized throughout the embryo. It's GFP tagged, and its concentration levels can be measured it's the amount of bicoid depends on the particular promoter that this fusion gene is fused to. There are promoters that drive relatively high expression or low expression characteristic of the most posterior regions of the embryo or middle regions of the embryo or high regions of the embryo. Then she used these lines to redo the chip experiments and to classify the chip experiments based on their binding under conditions of under in vivo conditions of different concentrations. So there are some lines that we call 143 of them bind what we call high affinity, and regardless of how low the bicoid, even the lowest bicoid concentrations, which is from the bicoid promoter itself, there's no change in the expression patterns in the binding or recovery of sequences. There are intermediate high lines, intermediate low lines, and low affinity lines. Now, we can take and ask for the biological meaning of this by going back to the Stark set, looking at the expression patterns of these different classes. And what you can see is that lines that are called high affinity and would be expressed even in regions of, if they're functional, and this binding is significant for predictive of its expression, should be expressed all in the posterior regions of their and that is true, as you can see, is this line here. And as one increases the affinity, one drives to increasingly anterior expression patterns. Interesting cases are actually from known genes where the enhancers have been characterized. So, for example, the giant GAP gene is expressed in 2 domains, an anterior domain and a posterior domain, 2 different enhancers, 2 different bicoid binding sites. The posterior enhancer is identified in the high affinity class. The anterior enhancer is identified in the low affinity class. We can take these pieces of DNA. What Colleen has done is to compare the apparent in vivo and in vitro binding properties of these genes. These genes separate in vivo in the embryo according to apparent affinities. But in gel shift experiments, we see no difference between anterior and posterior giant enhancers. And so what that suggests to us is that the property that's determining bicoid affinity is not strictly the DNA sequence, but some feature of that DNA sequence in the context of the embryo, in the context of normal chromatin. And two results that suggest that that's probably the case is that if you look at the regions which are regarded as show high affinity, they are highly enriched for sites that would bind chromatin factors, pioneer chromatin factors like Zelda that are thought to open up the chromatin. And so high affinity, and similarly, they also have, whereas low affinity sites have, in addition to reduced numbers of Zelda sites, higher general nucleosome affinities, as though they are more likely to be wrapped tightly with nucleosomes, such that the difference between high and low affinity sites is related to a chromatin feature which is we can think of as accessibility. And I won't go into those. And then interestingly, one we had also expected that potentially high affinity sites might have large numbers of bicoid binding sites or motifs, and the result is actually the opposite. That's the statistically significant behavior. Significant concentrations of bicoid binding sites are in the low affinity sites that would inherently have, we believe, inaccessible chromatin and are only become expressed because they have large numbers of bicoid sites which open up or make those sites accessible. So the model that we're proposing for bicoid, and we think this is an interesting model because potentially it's valuable or useful for, can be extended to almost all of the systems for, any system for concentration-dependent transcription factors that have multiple targets, is that the affinity of enhancers really depend on some chromatin accessibility features which may be inherent in the DNA but are not directly related singly to the bicoid binding sites. High affinity sites would be enriched for pioneer factors and moderate affinity for nucleosomes and moderate density of bicoid binding sites, and low affinity sites would be generally closed, and high concentration of bicoid would be required for getting accessibility. And we're beginning to test this model by choosing particular enhancers that have defined ratios of bicoid and Zelda or other pioneer factors, and switching them around and seeing if we can change enhancers from high affinity to low affinities. In general, those experiments have been successful. I'd like to end the talk, though, with stepping back again to the odd observation that although I spent most of my life focusing on genes that I know and gap genes that define or pattern were identified in our mutagenesis screen, there are, as I've argued, probably maybe up to at least 500 or maybe more significant functional enhancers, genes that are driven by bicoid. And all of these aren't part of an interactive network, I believe, and they probably define their boundaries are defined by interactions, direct interactions with bicoid. Now, the curious thing, if you look at this pattern, is some of the patterns are kind of what you would expect, but there are obviously other patterns as well. There are pair rule patterns, segment polarity patterns, odd subsets of pair rule patterns. In a way, this is surprising, but in a way, it's not surprising, because I think many of you remember the beautiful studies Steve Small and Mike Levine's lab on the Eve Strike 2 promoter, where in addition to gap gene binders, Eve, which is a pair rule gene, also has strong numbers of bicoid binding sites as well. And so we think this is probably a characteristic of almost all of these sites, that they have multiple controls, but bicoid is always one of them. Now, the question is, how does one think about that? How should one think about that? And one idea that I find intriguing stems from a theoretical paper by Michael Tikhonov, Sean Little, and Thomas Greger, where they point out that in terms of information theory, the standard way that I have always thought about information transfer of going to gap genes and then going from gap genes to pair-role genes and then from pair-role genes to segment polarity genes is a process that inherently loses information in that with each transmission or step in the process, you lose information that was initially present in it can only reduce information. This is very similar to this game that many of us have played as children, where you whisper something in someone's ear and they whisper it into the next ear and they whisper it. Every transfer loses information. And so in this paper, what they're suggesting is an alternate protocol where if you can, one of the best ways of achieving information or achieving precision is multiple readings of the same gradient. And so the alternate model that I'd like to suggest for thinking about gap genes and for thinking about pair-role genes, everything up to the final specification of patterning within the sentence, so that all of these expression patterns and other expression patterns, Bicoid's maternal control of caudal that feeds back on the system. All of these systems that we think of as feedback are not really interacting with each other. They're interacting with the downstream targets and represent multiple readings in this cell of the same Bicoid gradient, or that same Bicoid gradient over time. And it's that multiple readings that's possible from these complex enhancers that gives them their accuracy. Okay, very last point. Just triggered by this morning, I've enjoyed this whole talk so far. I think that it seems like I probably have to end this talk by pointing out to you that anterior-posterior patterning of fly embryos is not really like the kinds of complex interactions that occur in patterning in the neural tube. I think, and so then what one can do is say, well, strictly speaking, if there's a single message that I have to communicate with this in this talk, is the idea that, contrary to my own intuition, that biology isn't inherently sloppy, that gradients can be read accuracy over minute time scales that seem to preclude feedback adjustments, at least at the transcriptional level. And that certainly a system like this could work in a fly embryo where transcriptional responses are rapid, they occur without growth, and can establish position under positional accuracy in a straightforward way. So then the question is that the feedbacks, circuits, and the complex interactions that most of us are aware of that we work on in our other patterning systems, are they maybe really predominantly required only when fates have to be maintained over long periods, or particularly over long periods associated with growth. That the system can set up accurately, but it can't maintain patterns or requires patterns. Growth will certainly require the generation and interactions after the initial morphogens are away. I think the power of the Bicoid system may precisely be because it is so rapid that it is stripped down to simple biological mechanisms that might involve just transcription factor binding and output. And I think the usefulness of the system we anticipate over the next couple of years is the opportunity to actually understand how enhancers, how genes respond to concentration differences of that order. So I'm going to stop there. Again, the acknowledgements are two different sets of experiments. What I think of as the Big White Consortium, which is all my friends who sit around and who are mostly trained in physics and to a certain extent, but ever decreasing extent, have no idea about biology. But ever decreasing, I have to say. And also Colleen Hannon as a graduate student and Shelby Blythe in the lab, who did all of the in vivo measurements of big white targets. I'll stop there and take any questions that you have. Thank you.